Welcome to the chemical formulas and structures you should know for Biology 1110. We're going to start off with some basic side notes of what some of these symbols you're going to see today mean. C stands for carbon, N for nitrogen, O for oxygen, and H for hydrogen. When you see a single line, this refers to a single bond. When you have a single bond, one pair of electrons is being shared. When you see two lines, two pairs of electrons are being shared. This is a double bond. And in a triple bond, there are three pairs of electrons being shared. You may also see me write OH a great deal. Another way this could be written shows the bond between the oxygen and hydrogen. Finally, you need to understand the difference between a chemical formula and a chemical structure. A formula writes out the elements, such as H2O. The structure shows how they're connected to each other. The oxygen has two hydrogens bound to it. Now we're going to move on, and I want to get us started with our structure you're going to see quite a bit today. I'm going to go ahead and sketch this out for you very quickly. You'll see carbons, hydrogens, and oxygens. This one's actually a carbohydrate. You'll become very familiar with this semester. But I want us to spend a minute and count the parts. We have 1, 2, 3, 4, 5, 6 carbons. Hydrogens are next. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 hydrogens, and 1, 2, 3, 4, 5, 6 oxygens. Now this molecule is called glucose, and it is sugar produced by photosynthesis. Over here I'm going to draw a second molecular structure that I want you to know. We're going to get this stretched out. It too is made of carbons and hydrogens and oxygen. It is also a sugar or a carbohydrate. And we're going to count the pieces of this as well. And notice we have 6 carbons again. We again, surprise, have 12 hydrogens. And now for the oxygens. And you'll see we have 6. What's going on? It's the same formula and two different structures. One is glucose and one is fructose. When you have this occur, that is, you have the same chemical formula, but different chemical structures. There's a special name for this relationship between these molecules. They are called isomers. An isomer has the same chemical formula, but different chemical structures. Now we're going to move on to some molecular structures and formulas that you need to know. I'm going to start off just filling in the molecular formulas for you, and then I'm going to draw on the structures. I will provide a sheet that has these written that you can download. So if you will look, oxygen is made of 2 double-bonded oxygen atoms. Hydrogen, 2 single-bonded hydrogens. Carbon dioxide is a carbon double-bonded to 2 oxygens. Methane, carbon with 4 hydrogens. Water is oxygen with 2 hydrogens. Methanol is much like methane, but in place of that top H, you have an OH group. Ethanol has 3 hydrogens on your first carbon, 2 on the second one, and then an OH group. If you'll notice, both methanol and ethanol have OLs at the end of their name. This also goes with the OH found on their structures. These are alcohols, and that's how they can be recognized. Now for the carbohydrates, and there are 3 that I want to work on today: glucose, fructose, and ribose. Let me just get their chemical formulas jotted down here for you. Now, as we've mentioned before, glucose and fructose have the same formula, but different structures. I'm going to start off with drawing glucose. Again, it is a 6-membered ring. 5 of the members are carbon and 1 is oxygen. You have the final carbon branching off of the ring. So if you look at the overall structure here, I think it resembles a pot, the handle being the branched carbon, and in the center is the ring. Fructose has a 5-membered ring, or a pentagon. It has 4 carbons in the ring and 2 carbons branching off from the ring. This is how the structure differs from that of glucose. Remember, the formulas are the same on these two. They're isomers. Now we're going to move on to ribose. Ribose has a different formula. It is not an isomer of glucose or fructose. It is C5H10O5. You'll notice that it is a pentagon again, but only has 1 branch. So when you see a pentagon-shaped sugar, count the branches, count your carbons, and that will tell you if you're dealing with ribose or fructose. So please make sure you pay attention to that branch. And remember that both of these are what are called 5-membered rings, both ribose and fructose. Okay, so you saw how complicated those carbohydrates are. I'm going to show you a simpler way to draw structures such as glucose, fructose, and ribose, which are very carbon heavy and just end up very messy and complicated. This will work for pretty much all of your organic molecules. It relies on the fact that carbon and hydrogen are both implied in how we draw these structures. So here's glucose. Now, if you'll see here, I am drawing the same basic shape, but where I would have put my carbons before, I'm just leaving the joints connected. Then I have to write in my oxygens because they are not implied, only carbons and hydrogens are. So I'm going to fill in my oxygens and the hydrogens that go along with them as well. So now if you'll take a look at this structure, my carbons will be present at what we call the bends and ends of this structure. Everywhere aligned bends or aligned ends, which we do not have in this particular structure, a carbon is implied. So I'm going in here at each bend and drawing a red dot for the carbons. And now I'm going to write in the hydrogens. They are also implied. It is assumed that anywhere a carbon needs additional electrons to complete its outer shell, hydrogens are fulfilling that role. So now I'm going to draw the structure one last time, the nice clean way you could leave it, and you may see it on test or in other classes certainly. And there you have a much cleaner way of drawing glucose than including every carbon and hydrogen. And hopefully that will help you a little bit. Now for some additional structures I want you to be familiar with. One is glycerol, C3H5OH3. The base here is a simple carbon chain. Each carbon has an OH group coming off of it. And then all of carbon's remaining electron needs will be fulfilled by hydrogens. That is glycerol. Glycine is an interesting one. It is C2H5NO2. This one's formula is written oddly, so just bear with me. It has an amine group here on the end, your NH2, followed by some carbons, and then you have the acids, COOH, NH, you have an acid, and then you have hydrogens that fill in the middle. Glycine is an amino acid, which makes up a protein. Pyruvic acid, as you see the acid here I mentioned, the COOH comes into play again. This one is CH3COCOOH. You have a carbon chain. You have your COOH. You have a double bonded oxygen there in the middle instead of two hydrogens. Then you have lactic acid. Lactic acid is CH3CHOHCOOH. Again, you have a carbon chain here as your base, three hydrogens here on the end. You have an OH group coming off of your center carbon. Then you have your COOH, which indicates this is an acid. Now, if you'll bear with me for just a minute, I'm going to draw these in that simple structure we discussed before where the bends and ends indicate carbons. So there is glycerol. I'm going to do this for these two, and glycine. You must draw your ends. Nitrogen is never implied, just your carbons and hydrogens. Remember, carbon is found at your bends and ends, and hydrogens will fill any need for electrons that carbon may have. They're the three carbons in glycerol and our two carbons in glycine. Now, I'd like to talk to you about saturated versus unsaturated fatty acids. Right now, I'm drawing an unsaturated fatty acids. Like all acids, you have the COOH, but with an unsaturated fatty acid, if you look between your carbons, you have a double bond. This will help you recognize it as an unsaturated fatty acid. Unsaturated fatty acids are usually liquid at room temperature. Things like peanut oil, canola oil, and olive oil are examples of unsaturated fatty acids, and these are usually from plant sources. Now, a saturated fatty acid is a different story. Like an unsaturated fatty acid, it's a carbon chain. It does have the COOH on the end, but if you'll look, there are no double bonds between any of the carbons here. This signifies that it is a saturated fatty acid. Now, you may be asking, what is this saturated with? Well, the main difference here is the hydrogens between a saturated and unsaturated fatty acid. Let's count these carbons here in our saturated fatty acid, and you'll notice that both of these chains are the same length. And there are 8 hydrogens in our saturated fatty acid. In an unsaturated fatty acid of the same length, we have less hydrogens. Instead of 8, we have only 6. So a saturated fatty acid has been saturated with hydrogens. There are no double bonds present. Every hydrogen that can be crammed in has been crammed in, so there are no double bonds possible. Unsaturated fatty acids, on the other hand, usually always have double bonds present. A saturated fatty acid is usually a solid at room temperature because these can pack in tighter, and they are typically from animal sources when you think about lard. A plant-based source that you may have heard is Crisco. This, though, has had hydrogens artificially forced into it, and so that is what has made it a solid at room temperature. Unsaturated fatty acids are healthy. These are the ones that are better for you that your body can break down more easily. So, I'd like to start us now on nucleic acids, our last group of organic compounds. This includes DNA and RNA. They are composed of carbon, hydrogen, oxygen, phosphorus, and nitrogen. Their function is to control all cell functions or all cell processes. The subunit is called a nucleotide. A nucleotide is made of a sugar phosphate backbone combined with a nitrogenous base. Your nitrogenous bases that you will find in both DNA and RNA are adenine, cytosine, guanine. These three are found in both DNA and RNA. Now, found only in DNA is a nitrogenous base called thymine, and its counterpart in RNA is uracil. This is found in RNA only. Now, for that sugar phosphate backbone, I'll represent the phosphate portion by a P with a circle around it, and your sugar is a 5-membered ring. This will be your 5-carbon sugar. There is a branch that comes off of that. And this is how we're going to draw these. You have your phosphate connected to your sugar, phosphate to sugar, phosphate to sugar. This is what makes up the backbone. And I'm going to draw a very short backbone here. This will be much longer than an actual piece of DNA or RNA. Off of each sugar you have a base. These are your nitrogenous bases that I mentioned before. So here are all of your nitrogenous bases. These will vary. You may have, they are represented by an A, T, C, U, or G. And this is your sugar phosphate backbone. And this is the basic structure of both DNA and RNA. Now, there are a few differences between DNA and RNA. I only want to mention a structural one here. DNA is composed of a double helix. Nitrogenous bases match up with each other and are held together with hydrogen bonds. RNA is a single strand. The bases come off the sugar phosphate backbone. So you have a double strand in DNA, also called a double helix, and a single strand in RNA. We'll cover the other differences later in the semester. Thank you for watching this video on the atomic structures. I know it's a lot of information to take in. I'm sure you're feeling rather overwhelmed. Please don't panic. Watch the videos on each of these groups individually for details about the different types of organic molecules, and download the sheet Structures to Know from eLearn. It has a list and drawings of each of these molecules that you're responsible for, but I know sometimes it helps to have someone walk you through each of these structures. You should be able to recognize these in a number of ways, including the structural formulas that I've been drawing here, the simple bends and ends diagrams, and you should be able to recognize them as you see on the screen here with the balls. The red represents oxygen, the black carbon, the white hydrogen, and blue is usually represented by nitrogen. So get comfortable with these structures, be able to recognize them, know what formula goes with what structure, be able to look at a structure and be able to write down the formula by looking at the structure, be able to recognize glucose and fructose, so that they are isomers, know how they differ from ribose, know how they differ from one another. Work on being able to recognize these structures and pick them out of the group. Good luck, and please let me know if you have any questions.